So far, out of all the biological molecules, we had a chance to discuss carbohydrates and lipids. Today is the turn of amino acids that are building blocks of proteins. Now, this statement right here, that proteins are among the most prevalent types of molecules in living organisms, is probably a bit of an underestimation. If you think about the function, any function in a cell, 99% chance this function is carried out by proteins, whether it is a structural function, signaling function, or catalytic function. Now, all proteins are composed of amino acids. So within the human body, we count 20 amino acids. Now, I want to make a sort of a I need to clear my conscience. If you would look at the composition of all human proteins, and I mean all of them, there will be 21 amino acid. I will mention the 21st one. That 21st amino acid was discovered by the scientific criteria relatively recently. It was discovered in early 1990s and was extensively studied since then. Now, the function, the structure, and the function of the protein is determined by the order of the building blocks, the amino acids that compose it. And functionally, proteins can be structural components. Muscles, hair, cartilage are all formed by proteins. They can function as enzymes. Remember, when we were discussing the rate of the reaction, we mentioned that there's such a thing as a catalyst. Enzymes are biological catalysts that accelerate the reactions in the living cell. A lot of the proteins serve as signaling molecule. Some of them serve as transport molecule. And this table here summarizes main functions of different types of proteins. For instance, collagen and keratin form connective tissue such as tendons, ligaments, and cartilage. On the side of collagen, and keratin forms hair and skin. Some proteins allow us to move. Contractile proteins like myosin and actin are essential for the muscle contraction. Transport proteins such as hemoglobin or lipoproteins can move substances. Hemoglobin transports oxygen in the blood, and lipoproteins transport, as you may remember from this Monday's conversation, various types of lipids from cholesterol to fatty acids, from liver to tissues and back. There are storage proteins. So, for instance, casein is the most common protein found in milk. If you're probably familiar with cottage cheese, right? The curds in the cottage cheese, that's the denatured protein casein. Ferritin is an extremely important protein in the human liver, which binds and stores iron. A lot of proteins serve as hormones, from insulin to growth hormone to antidiuretic hormone to a variety of hypothalamic hormones, the old proteins. Of course, one of the main functions of proteins is to catalyze biochemical reactions. They serve as enzymes. Example, enzyme sucrase breaks down sucrose, the table sugar, into glucose and fructose. And trypsin breaks down a variety of proteins. As a matter of fact, you can physically sense the action of one of the protein enzymes that are in your saliva. I think I mentioned that. The salivary amylase, the protein enzyme, breaks down starch into individual glucose molecules. So if you will munch on some bread and keep it in your mouth for several minutes, you will start feeling the sweet taste because the starch will be broken down to glucose, and glucose will trigger the corresponding response. Okay? And of course, proteins serve as the protection. Immunoglobulins, also known as antibodies, are proteins that protect you from infections. The building blocks of proteins are amino acids. And what is interesting about amino acids is that the large chunk of their structure is the same. So all amino acids have a central alpha carbon. All of them have the ammonium group and the carboxylate group. Let's take a look where these groups are on this image that I have on the slide. So if you look at the structure of the amino acid, any amino acid, there will be an ammonium group here, okay? You can see that nitrogen, which normally forms 3 bonds, forms 4 bonds here because it accepts an extra hydrogen, which gives this group a positive charge. Now, carboxylate group, which usually is depicted like this, at the physiological levels of pH, loses hydrogen, becoming negative, and that hydrogen is the one that is accepted by the amino group. So you can see that amino acids, they have 2 charges. They have a positive charge on the ammonium group and the negative charge on the carboxylate group. Does that make sense? Overall, they are neutral because the positive and the negative charge cancel each other. But they do have this distribution of charges across the molecule. Again, at the physiological pH, the pH of 7.4. The carbon atom, in between them, is called an alpha carbon. And these 3 elements, alpha carbon, carboxylate group, and the ammonium group, are found in all amino acids. Now, this R group, or side chain, is what makes amino acids different. All right, so let's take a look how it happens. Based on the structure, so since ammonium group, carboxylate group, and alpha carbon are common for all amino acids, we should distinguish them based on the structure of the side chain, or R group. So broadly speaking, all amino acids can be divided into nonpolar and polar. Nonpolar amino acids have hydrophobic side chains, which for the most part consists of hydrocarbons. Hydrophilic amino acids, on the other hand, have polar side chains or ionic side chains that either contain very strongly electronegative oxygen and nitrogen, or even charges. Here you see 2 examples. You see the amino acid valine, which side chain contains only carbon and hydrogen atoms, meaning that it contains essentially hydrocarbons. And polar amino acid asparagen, which side chain contains highly polar carbonyl and amino groups. So far, so good, everyone? Okay? So now, I'm going to preface it. Will you need So will you have a question on the exam 4 to identify polar, charged, or nonpolar amino acids? The answer is yes. Will you have a table with amino acids? The answer is yes as well. Do you understand what I'm saying? So I will provide you with the structures of all 20 amino acids. So if the question states, which of the following amino acids is polar, and you have a list, you can always refer to that table with all the structures and figure out which one is polar and which one is not. Good? Let's take a look at non-polar amino acids first. Glycine and alanine are perhaps two simplest amino acids. You can see that glycine has a hydrogen atom as a side chain as an R group, and alanine has a methyl residue, so they are nonpolar. Valine, leucine, and isoleucine are fairly similar in their structures. They all are nonpolar because you can see that their side chains are essentially nonpolar hydrocarbons. So is the side chain for an amino acid called phenylalanine. If you will compare the amino acid alanine to phenylalanine, you can see that the difference between the two is that one of the hydrogens on the side chain of alanine is replaced by the phenyl substituent. Nevertheless, phenylalanine is still nonpolar amino acid because it contains only carbons and hydrogens in its side chain. So far making sense? Now, I'm going to skip methionine for now. So proline is one of kind of unique amino acids. Do you see the 5-member cycle that it forms? It's a very interesting amino acid. It's relatively rigid compared to others, which can be very important in terms of the protein structure and the protein function if proline is found somewhere in critical place, like an active site of an enzyme or something like that. Now, what about methionine? So methionine contains that sulfur atom. Basically, it's a thioether. But the difference in electronegativity between sulfur and carbons isn't that huge. And on top of that, if you think about this, sulfur will be attracting electron density towards itself. It is more electronegative than carbon. We don't have the periodic table in the class, but I can assure you it is, okay? So these bonds are slightly polar, which means each of these carbons has slightly positive charge, and sulfur has slightly negative charge. But since it's a linear molecule, these positive charges and this negative charge basically cancel each other out. There is no dipole. There is no uneven distribution of charges. So methionine is another example of nonpolar amino acid. And finally, tryptophan, okay? Despite the fact that it contains this nitrogen atom, it has to be polar. The structure of tryptophan allows it to distribute. So you see there are a lot of carbons, carbon, 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 carbon. All of those are carbons. So even the presence of more electronegative atom, which is nitrogen, does not affect the polarity of this large side chain to the degree that tryptophan becomes polar. It is still a non-polar amino acid. Now let's take a look at the polar amino acids. So what do you have to have within the side chain for an amino acid to be considered polar? They got to have hydroxyl, thiol, or an amide group. So let's highlight the functional groups that we see in the following amino acids. They're polar. Serine, threonine, and tyrosine all have hydroxyl groups. Okay? Cysteine has diol, which also makes this sulfur-carbon bond here, highly polar. And asparagine and glutamine have the amide group. So you may remember, I'm going to remind you, when we were talking about carboxylic acids, we mentioned that this is the typical carboxylic acid. If you replace OH with NH2, it's an amide. Okay, so far so good. Now, these are examples of polar amino acids which do not carry any charge. There are amino acids that do. They can be either acidic or basic. Remember, acidic amino acids, well, acid, according to Bronsted-Lowry. Remember Bronsted-Lowry theory? Acid donates hydrogen. Base accepts hydrogen. So, both aspartate and glutamate, they donate hydrogens, which makes them acidic. So they're not just polar, their side chains are also negatively charged. Make sense? And there are 3 amino acids which can serve as Bronsted-Lowry bases. Histidine can accept the hydrogen. Lysine can accept the hydrogen. And arginine can accept the hydrogen. When they accept that hydrogen, they gain positive charges, which makes them charged and polar. Now, before we move on, I want to remind you something. I want you to kind of keep in mind that if something is polar, it's hydrophilic. It readily interacts with water molecules. If something is nonpolar, it's not hydrophilic and it doesn't interact with water molecules. All that. We good, everyone? Moving on? Okay. So, there are multiple ways to represent amino acids chemically. Obviously, you can draw a structural formula. So it will contain the alpha carbon, the ammonia group, the carboxylate group, and the side chain. But often, especially when you need to talk about polypeptides or proteins in which you have many amino acids, drawing the structural formulas becomes a bit of a nuisance. So what we do, we use either a 3 or 1 letter abbreviation that is kind of derived from the name for an amino acid. So, okay, let's take a look at these 2 amino acids here, asparagine and aspartic acid. And let's identify some of the parts of it. So what I'm going to do, I'm going to highlight different parts of the amino acids. And you will tell me what we are talking about. What is this? the atom that I highlighted, which is alpha carbon, right? Make sense so far? What is this? That's the ammonium group, yes, because it's protonated, so we call it ammonium group. Okay? What is this? What do we call it? So, sorry, ammonium on the left, on the right, it's going to be what? The C word. Carboxylate. And finally, you see the side chain, right? It's already shaded for us. Or R group. So again, I want to stress it that amino acids are different because of the side chains. Okay? This is what makes them different. Well, let's take a look. We're going to try to identify these amino acids as being polar or non-polar. So first one is glycine. I can say that this carbon, I'm going to kind of kill it. This carbon is connected to 2 hydrogens. Glycine, is it polar or non-polar? Do you need to look at? Non-polar. Why? You're right. It doesn't have any polar substituents in the side chain. What about threonine? It is polar. Which substituent suggested this answer? Yes. So hydroxyl group says that it's polar. Hydroxyl, thiol, or amide. And indeed, right. So, here's the thing. Obviously, you will not have to draw anything during the exam. You may need to select the structure, appropriate structure. So, let's do valine. First of all, there are common elements in each amino acid. Remember, alpha carbon, ammonium group, and carboxylate. So here's our alpha carbon, which is connected to 1 hydrogen. It is connected to ammonium group. How many hydrogens at the physiological pH should we have at the ammonium group? 2 or 3? 3. And the charge will be 1 positive. Your carboxylate group is going to lose hydrogen because it acts as an acid at the physiological pH, and it's going to have 1 negative charge. Does that make sense? Actually, let me do something here. So, I'm going to do like theoretical part. So this is an absolutely hypothetical amino acid. R represents the side chain. Do you follow my train of thought? At the physiological pH, you can think of it this way. This hydrogen is transferred here. When hydrogen is transferred, it is positively charged. Am I making sense? So what you get, when carboxylate group loses the positive charge, it becomes charged negatively. When NH2 receives an extra hydrogen, it becomes charged positively. Sounds good? Now, in this particular example, we're talking about an amino acid, valine. It's a nonpolar amino acid that looks like this. Again, I despise the idea of memorizing all formulas for amino acids for no particular reason. I doubt that anyone here, including me, will ever need to know all 20 amino acids and all of their side chains. Like seriously, okay? Make sense? But you should be able to identify if it's polar, if it's non-polar, and basically recognize an amino acid in the table. What about cysteine? Cysteine is the polar amino acid. And again, it's going to have the same common structural elements as any other one. Making sense so far? But the side chain will be different. So in cysteine, the side chain will be this. Okay? Does that make sense? Again, I know it. It's not that, oh my God, how do I remember? You look in the table. Basically, you Google it, you know? I'm not paying too much attention to memorizing all the structural formulas. Before we move on, does that make sense? So again, amino acids differ because of the different side chains, but they also have common structural elements. The carboxylate group and the ammonia group. So these amino acids can be linked together to form polypeptides. When two amino acids are linked, form peptide bonds, which connects the carboxyl group of one amino acid and the ammonia group of another. You can see the peptide bond from the illustration to the right. So, if you take two amino acids and link them together, we call it a peptide. And specifically for two amino acids, it's going to be dipeptide. 3 amino acids, tripeptide. 4, tetrapeptide. You can go on. You can say 5, pentapeptide, 6, hexapentide, and so on and so forth. You know, like you can move on. At some point, you just start calling it a polypeptide. Okay? So, let's take a look at the formation of the peptide bond. So, you have this two hydrogens and one oxygen. You see that the oxygen belongs to the amino acid on the left, glycine, and hydrogens belong to the amino group of alanine on the right. So two hydrogens and oxygen leave these molecules, forming water, and carbon and nitrogen become connected, forming a peptide bond. Good? Before I'm going to say something really important, like seriously important. And you can keep going. You can keep adding more and more amino acids on either end. But I want to stress something really important about those peptides. Even in this dipeptide, you can clearly see that the ends of it are different. Can you see that? So there's one end of this two amino acid peptide, the dipeptide, which contains ammonium, nitrogen atom. Does that make sense to you? We call it N-terminus. And there is another end which contains carboxyl group. We call it C-terminus. Every protein, every polypeptide has an N-terminus, the end where there's an ammonium group, and the C-terminus, the end where there is a carboxylate group. So this is just a fancy way to represent what happens when glycine and alanine form a peptide bond. But it doesn't differ from the structural formulas that we saw a couple of slides ago. Same way the water leaves the reaction mixture, and you have a peptide bond formed between the glycine and the alanine. Now, how do we name them? Remember, it is something that I try to instill in you folks. A lot of the stuff that we talk about, like how we name things, how we classify things, it's artificial. Like, we decided. You understand what I'm trying to say? We can say, oh, how many males versus females in the class, but I can classify students, okay, how many students wear black pants and how many students wear not black pants. Also classification, useless, but it's classification. So, naming conventions are also sort of an agreement. So the naming convention is this. The amino acid that is located at the C terminus retains its full name, alanine. Amino acids that are closer to the N terminus go from, let's say, glycine to glycyl. Am I making sense? All good? So for instance here, if you don't mind, I don't want to draw it because people who prepared the slides already did that. Basically what you need to do, you need to take an oxygen from here and 2 hydrogens from the ammonia, right? So what you're going to get, so I can say that minus, so water leaves, and now you have the peptide bond. Does that make sense to you? So what I highlighted with the yellow in that dipeptide is the peptide bond. Now, the full name for each amino acid, let's take a look. This is serine, and this is threonine. So let me ask you this, which amino acid is on the C terminus? Remember, this is the C terminus. This is the N terminus. So it's a threonine that's on the C terminus. Can you see that? So it retains its name. What happens to the name of the amino acid on the N terminus? We replace INE with YL. Serial threonine. Does that make sense to you? We're going to get some more practice pretty soon. So here's the idea. The C terminus retains, so it's serine, it remains serine. All other amino acids change from the proper name to the root with YL. So glycine becomes glycyl, alanine becomes alanyl, and the full name is alanyl, glycyl, serine. You read from N to C terminus. Again, it's a convention. Does that make sense to you? So the name, we have 4 amino acids. Which amino acid is on the C-terminus? Methionine, so it retains its name. What do we do with cysteine? Cysteine. Leucil. Alanine. Okay, so here's the deal. Listen up. We have 4 amino acids. We have tetrapeptide. By biological standards, it's a small molecule. And now we have the name. Alanine, leucil, cysteine, methionine. That sounds like German. Like seriously. That's a long name. And if we keep adding amino acids, it's going to get longer and longer and longer. And eventually, imagine you have a I believe human insulin is like 51 or 57 amino acids. Yeah, and it's a small one. It is a small protein. The spike protein of SARS-CoV-2 that is used in vaccines, 1,262, I believe. Yeah. Writing all that name will take you like half a day, right? So this is why we use 3 and 1-letter abbreviations. So 3-letter abbreviations here would look like this. Alanine, leucine, cysteine, methionine. Okay? For these specific amino acids, single letter abbreviations would match the first letter. So it will be A, L, C, M, but nobody puts the hyphens between the letters, so the peptide will be like this. Making sense? The table with amino acids will contain the 3 and 1-letter abbreviations. So that's what we just wrote. Now, we get all the amino acids from proteins. And we can synthesize 11 of them. And 9 amino acids that are shown here, we can't. Okay? Sounds good? We call them essential amino acids. And if you consume animal protein, you're all covered. Because you receive animal protein, whether it comes from chicken, fish, beef, pork, you name it, milk, butter, well, butter is mostly fat. All of those products contain all essential amino acids. However, if you're a vegetarian, your diet doesn't contain. Well, it can contain, but here's the deal. If you decide that you're going to go as vegetarian and you're just going to eat only grains, or only beans, or only nuts, you're not going to get all essential amino acids. You still can get them, you just need to combine all these products together. Does that make sense? It's been shown, it is known that the diet, vegetarian diet, that consists of the combination of legumes, basically beans, vegetables, grains, nuts, provide you with all essential amino acids. Sounds good? This is why, by the way, it's not like vegetarian diet is not recommended for very young children, well, children in general, because they require an enormous amount of proteins to grow. And it's really hard to supply the necessary amount of amino acids to a growing child using a vegetarian diet. Like seriously, it is insanely hard. Okay? It's just much easier with animal protein. Any questions so far? Okay, let's talk about the protein structure. This is actually pretty cool. So Proteins are highly organized molecules, and their structure is guided by the interactions between amino acids that comprise them. The first level of protein structure is the primary structure. This is really simple. Primary structure, by definition, is the sequence of amino acids. So this tetrapeptide that you see on the screen, its primary structure can be described using these three-letter abbreviations. Okay? Make sense? Now, I want you to acknowledge something. There are a lot of polar groups. Carbonyl, amido, amino, carboxyl. Do you see that? They are polar, these groups. So interaction between these groups, as well as interaction between side chains, will determine the next levels of protein structure. So this is an example of thyroid hormone, okay? So I really don't like when chemists start to, get their dirty hands into the material that they don't know. So this is not a thyroid hormone, okay? Let me correct some stuff here. So thyroxine is the thyroid hormone. What they refer to here is the thyroid stimulating hormone. So it's not. It is a hormone that is produced by your pituitary gland. It stimulates your thyroid gland located in your neck to release thyroxine. Okay? Here's what's important. Check this out. So you see that there's 3 amino acids. We have an amino acid glutamine, histidine, and proline. Okay? Now, I'm going to give you a bit of a challenge. It's not complicated. We have 3 letters, G, H, and P. E, H, and P. Everybody agrees? Let's put them in as many combinations as possible. So we can have this, right? Can we have some other combination? Can we do this? Yes? Sure, why not? Can we do this? What about this? Right? I don't know if I ran out of the options, but it looks like I'm kind of getting there, okay? Yeah, I think that's it. So you can take 3 amino acids and put them in a variety of combinations, but the only biologically active combination is this. Why I use this example? It exemplifies the importance of the primary structure of the protein for its function. If you rearrange amino acids in the different order, it's going to be different. Okay? Does that make sense? It's like letters in the word. Okay? Am I making sense? Right? Okay. So the first protein, which primary structure was determined, was insulin. It was a massive feat. I don't remember the name of the dude who did that. I don't remember. A Nobel Prize for sure. Okay? I once checked how many Nobel Prizes, quote unquote, insulin won. There's like 4 or 5 Nobel Prizes associated with insulin. The structure, the discovery, the synthesis, all of that. Okay? Now, this is real exemplary. So insulin, in fact, consists of 2 chains, A and B. Can you see them? Each chain has the certain order of amino acids, and they are connected by so-called disulfide bonds. If you will look closer on the disulfide bond formation, you will notice that the amino acids that form disulfide bonds are cysteines. Am I making sense? So what exactly happens there? Well, check this out. You have 2 cysteines. I'm just going to produce like sort of schematic description of this. So this is one cysteine. And there is another cysteine here. Sounds good? So what happens? They get oxidized. These hydrogens are removed, and 2 sulfurs are now connected by a covalent bond. So they are chemically attached to each other. This property of cysteine is an extremely important feature in terms of structural organizations of proteins. Okay? Good? So let's answer some questions here. First of all, where is the C terminus? On the right or on the left of that polypeptide? On the right. And N-terminus is obviously on the left. Now, if that would be an exam question, what would you do to identify individual amino acids? What would you use? I would give you a table, right? So first amino acid, remember, it has to start with a nitrogen and end with a carbon. So that's your first one. This is your second one. And this is your third one. Am I making sense so far? So these are alpha carbons, and these are side chains. We're not gonna like go to the table and search for them, because at least I know these ones. So the yellow amino acid, the one on the C terminus, is alanine. You need to look at the side chain. The amino acid in the center is cysteine, and amino acid on the N-terminus is phenylalanine. Are we good so far? So the name will be, how should I change phenylalanine? Reminder, phenylalanyl. How should I change cysteine? Cystyl. Do I need to change alanine on the C-terminus? I do not. Just for the practice, the 3-letter abbreviation, phenylalanine, cysteine, alanine. I'm ashamed to admit that I do not remember the one-letter abbreviation for phenylalanine. I remember that it's, I want to say it's F, but I'm not entirely sure. Okay? I never remember them. Like, there are some oddballs there. Okay? So, but yeah. We've got it. So a lot of the signaling molecules and neurotransmitters are polypeptides. Okay, so this is an example. There are enkephalins and endorphins. And I just want to make a bit of a case here. So this is the beta endorphin. It's a pretty long one. Okay? The enkephalins are much shorter. They're pentapeptides. But what's common between two of them is that both of them have painkiller activity. In your brain, there are, and mine too, there are opioid receptors. Does that make sense? Both endorphins and enkephalins can bind to those receptors, and they can suppress pain sensations, and they also give you, like, you feel good. Enkephalins and endorphins are largely produced when you exercise and are responsible for exercise high. It's a normal term. Now, the term opiate should sound familiar, shouldn't it? Right? So, it emphasizes the sort of a primate of structure over the sequence. So structurally, opioids, such as morphine, heroin, fentanyl, hydrocodone, OxyContin, whatever, they bind to the same receptors, but better, and stimulate them stronger. Does that make sense? So the whole painkiller activity of synthetic or natural opioids is due to the binding to the same receptors that these two guys bind to in your brain. Okay? Am I making sense? We'll get to oxytocin and vasopressin. That make sense? And the biggest problem that is with opioid addiction. I mean, these molecules, they stimulate opioid receptors. But synthetic opioids stimulate it way better. And eventually those receptors become desensitized. They become resistant. Am I making sense in what I'm saying? So basically, it's like if you work at the construction site without ear protection, you eventually become deaf. So those receptors become essentially deaf, and they need more and more stimulus to actually act. Okay, does that make sense? And this is why if you have someone who suffers from addiction to opioids and you take them off the opioids cold turkey, one of the side effects is pain. They're so used to using these highly effective drugs that their own endorphins and enkephalins fail to adequately stimulate the receptors. Am I making the receptors, like, they don't care? Good news, the sensitivity of the receptors can be restored. So people who quit the synthetic opioids, eventually the sensitivity of the opioid receptors in them, it becomes greater, and enkephalins and endorphins, they start working. But initial period is really complicated. Okay? Another example of biologically active polypeptides are oxytocin and vasopressin. So these are very interesting polypeptides. We're going to do the learning check and I'm going to kick you out. So oxytocin is the hormone which regulates the delivery in females. Okay? It stimulates contractions of the uterus. And if labor has to be stimulated, it's usually oxytocin that is injected intravenously and stimulates uterine contractions, and the labor starts, okay? It's not produced by pituitary gland. Again, this is my despise towards chemists. It's not produced. It is produced by the organ called hypothalamus, but we're not in the physiology. It is released. It is stored in the pituitary gland and released from it into the circulation when needed. Vasopressin is a really cool hormone. It regulates how much water you're going to pee out. Now, the name for this is antidiuretic hormone. So if you are dehydrated, this hormone will keep water inside. Sounds good? Reasonable, right? So you will release very little water. Your urine will be low volume and very, very concentrated. If you overhydrated, the opposite. Making sense? Like you drink a lot of fluids, your urine is really diluted, and you pee a lot. So here's my question. So imagine you drink 3 pints of water. Are you going to run to the bathroom, like one after another? Are you going to run to the bathroom pretty soon? Yeah. So you replace water with beer. Same 3 pints. You going to run to the bathroom? Yeah, I think so. You replace beer, not with the same volume, but the same alcohol content, but instead of beer, drink vodka. Basically, you take 3 shots of vodka. Volume-wise, it's very small. But you're going to go to the bathroom really quick. Alcohol inhibits the actions of vasopressin. It doesn't work anymore, and you keep excreting water. Does that make sense? One of the effects in the morning, if you abuse alcohol too much, is dehydration. It's one of the main components of hangover. Okay? Just a little tidbit of how our knowledge about the function of those hormones can explain certain life events. Now, tripeptides. Okay, names. Let's do this. We have 2 glycines and 1 alanine, right? So G, G, and A. Sounds good? How can we combine them? Tell me. 3 letters. How can we combine them? Okay, let me start. If you're so shy. GAG. Works? Give me another alternative. Perfect. And the third one? GGA. Remember, peptides have a direction. So this is N-terminus, this is C-terminus. Conventionally, N-terminus is on the left and C-terminus is on the right. Just, that's an agreement. Am I making sense? If you want to name them, I'm just going to say it out loud. So the first one will be glycyl, alanyl, glycine. The second one will be alanyl, glycyl, glycine. And the third one will be glycyl, glycyl, alanine. This is your solution. Okay? Good? So next time, probably on Monday, I hope to not finish, but do a chunk of secondary, tertiary, and quaternary structure, and maybe discuss some enzymes.